In 2008, a spacecraft flew through a rain of ingredients for life. It collected everything. It recorded everything. And nobody looked at the data for 17 years. That spacecraft was Cassini. And what it was flying through were the geysers of Enceladus, a moon of Saturn, roughly the size of the United Kingdom, covered in white ice, that permanently ejects columns of material hundreds of miles into space. Fountains from another world, active for millions of years, visible from Saturn's orbit to any instrument passing nearby. Cassini flew through those geysers at 11 miles per second. A rifle bullet travels at about half a mile per second. Cassini was moving 20 times faster than a bullet through a fountain of water and ice rising from the interior of another world, just 13 miles above the surface, collecting every grain it passed through, recording every chemical signature with its instruments. Mission complete. Files archived. Move on. For years, those data files sat there, stored. Nobody deliberately ignored them. It was simply that the analysis tools of that era had limits. Complex molecules leave subtle chemical signatures in mass spectra. In 2008, some of those signatures were technically detectable but buried in noise, below the detection thresholds of available methods. The instrument on Cassini that collected this data is called the CDA, the Cosmic Dust Analyzer. It was designed in the 1990s to analyze dust particles and ice grains. It did its job. It recorded everything. The problem wasn't the data collection, it was the interpretation. And interpretation improves over time, with better methods, with researchers asking different questions than the ones being asked when the mission was running. October 2500. A research team publishes a paper in Nature Astronomy. They had reopened the data from that flyby, not to find something specific to look more closely, using analysis algorithms and spectrometry models that didn't exist in 2008. And there, inside files that had been sitting on archive servers for 17 years, they found something nobody had seen. Esters, alkenes, ethers, nitrogen and oxygen-bearing compounds. Complex organic molecules, directly from the subsurface ocean of Enceladus, ejected into space inside freshly formed ice grains, collected by Cassini 13 miles above the moon's surface. Those grains hadn't had time to recombine with other compounds in orbit. They had come straight from the interior, intact a raw chemical signature of what was happening at the bottom of that ocean. You know esters without knowing it. They are what gives fruit its smell. The characteristic scent of a ripe banana, a strawberry, a pineapple that's an ester. Those same molecules, not an equivalent, not something vaguely similar, those exact chemical structures have just been identified inside geysers on a moon of Saturn, 870 million miles from here. These are not simple molecules. Esters don't form by accident in cold water. They require specific chemical reactions to exist: heat, organic precursors, particular conditions. Their presence in those ice grains ejected from the interior of Enceladus doesn't say there is life there. But it says something equally important. The ocean beneath that ice is not a dead stretch of water. It is a place where complex chemistry is actively happening, right now, on a moon 870 million miles away. The answer to the greatest question in human history, are we alone, was possibly sitting in archived files for 17 years. Nobody had looked in the right place. And that's not a metaphor. These are files on servers, accessible since 2008, with the answers inside. And that's not all, because what Cassini flew through in 2008 Enceladus is venting it into space right now. What Enceladus is shooting into space right now, as you're watching this, Enceladus is ejecting water, ice, and these organic molecules hundreds of miles into space. These are not traces of past activity, not the remnant of a long-gone era. This is active, continuous, happening now. Enceladus's geysers have been running permanently, probably for millions of years, projecting material from the interior of the moon directly into space. Some of those plumes reach over 300 miles in height. The ejected material ends up feeding one of Saturn's rings, the E-ring, which is directly made of particles that came from inside Enceladus. Think about that. You look at Saturn's rings in a photograph, those iconic structures that make Saturn the most recognizable planet in the solar system. The E-ring is not primordial rock left over from the birth of the solar system. It is material from the interior of a moon, ejected into space by active geysers, dispersed into orbit around Saturn. And inside that material, since October 2005, we know there are complex organic molecules. Part of Saturn's rings is made, at least in part, of the same chemical building blocks found in living things. The geysers exit through Enceladus's south pole. That is where the ice crust is thinnest, between less than a mile and 3 miles thick, compared to 12 to 15 miles elsewhere on the moon. You can see the fissures from space, dark lines nicknamed the tiger stripes. They are not passive scars. They are active conduits. The pressure of the subsurface ocean searches for a release point, a weakness in the ice crust, and finds it there at the South Pole, where the ice is thin enough to give way. That is not a geological coincidence. It is the signature of an active ocean, under pressure, pushing from below and finding its way out into space. What Cassini flew through in 2008 was exactly that, water and material from the interior of a subsurface ocean, forcing its way through cracks in the ice and shot hundreds of miles into space at -328 degrees Fahrenheit at the exit, while below, under 15 miles of ice, the water stays liquid and active at above 19.5 degrees Fahrenheit. That contrast -328 at the surface, above 19.5 at the bottom, says everything about what Enceladus actually is. A world that looks frozen, dead, hostile. A world hiding inside it a warm, active, chemically complex environment shielded from space by an armor of ice. What makes this even more staggering is the timescale. Enceladus has probably had this ocean for hundreds of millions of years, possibly billions. The hydrothermal vents at the bottom of that ocean have been interacting with water, minerals, and organic molecules for a span of time the human mind cannot fully grasp. On Earth, the earliest traces of life go back roughly 3.5 billion years, and the oldest microbial fossils suggest that life appeared quickly after habitable conditions were established. Enceladus has had those conditions for at least as long. What has happened there during all that time, we don't know. We have never looked. To understand how any of this is possible, you have to go down. The hidden ocean and the fires below in 1977, we believed all life needed the sun. That year we discovered we were wrong. And that discovery is exactly why Enceladus is now the #1 priority in astrobiology. Beneath Enceladus's frozen surface, there's a global ocean. On a localized pocket of water somewhere under the South Pole, an ocean that wraps around the entire interior of the moon under an ice shell averaging 12 to 15 miles thick. That ocean is roughly 25 miles deep, 6 times deeper than the lowest points of the Indian Ocean. And at the bottom of that ocean, hydrothermal vents at temperatures above 19.5 degrees Fahrenheit. Here's what we believed before 1977. Biology had an absolute rule. All life needs energy, and energy comes from the sun. Plants capture sunlight and convert it into organic matter. Herbivores eat the plant. Predators eat the herbivores. Trace any food chain on Earth back far enough and you always arrive at the same source. The sun. It was self-evident. An unquestioned scientific consensus. No sun, no energy, no life. In February 1977, a research team aboard the vessel Nora was exploring the Galapagos Rift at 13,000 feet of depth in total darkness. They lowered a camera, and in the images that came back up, they saw something they didn't immediately understand. Entire colonies of living organisms, giant tube worms over 3 feet long, white crabs, translucent shrimp, mussels clustered around cracks in the ocean floor from which mineral-rich water was venting at temperatures exceeding 750 degrees Fahrenheit. No sunlight whatsoever. No photosynthesis possible. That day, the definition of life changed. These organisms were using chemosynthesis. Instead of converting light into energy, bacteria living within their tissues were converting hydrogen sulfide, a gas toxic to us, into usable chemical energy. The entire food chain rested not on the sun, but on the heat and chemistry of the ocean floor. A complete, self-contained ecosystem, totally independent of the nearest star. And the immediate question, the question every astronomer and every biologist asked from that moment on was, if life can exist without sunlight at the bottom of our own oceans, where else can it exist? Enceladus checks every single box. A liquid ocean, kept warm not by the sun but by the tidal forces Saturn exerts on the moon, gravitationally kneading it for billions of years. Saturn is 95 times more massive than Earth. The gravitational force it exerts on Enceladus deforms the moon, continuously compressing it, releasing it, compressing it again, orbit after orbit. That continuous deformation generates heat through friction inside the moon itself, exactly the way your hands warm up when you rub them together. This is called tidal heating, and it is what has kept Enceladus's ocean liquid for billions of years, 870 million miles from the sun, where without this internal heat source, everything would have been frozen long ago. Active hydrothermal vents at the bottom of that ocean. Complex chemical compounds in the water. A thick ice shell acting as a thermal insulator, keeping the heat inside and shielding the environment from space radiation. Scientists found direct evidence of those hydrothermal vents in Cassini data well before the 2025 discovery. Silica nanograins, microscopic particles that form when hot water interacts with rock at temperatures above 19.5 degrees Fahrenheit, had been identified in Enceladus's plumes in a 2015 study. These grains can only form under one specific condition: direct contact between liquid water and silicate rock at high temperature. Their presence in the geysers proved, before any other discovery, that at the bottom of that ocean there was something resembling the hydrothermal vents of Earth's deep sea floor. Enceladus is, to this day, the only place in the solar system beyond Earth that simultaneously meets all 4 conditions: liquid water, an internal chemical energy source, minerals, and now complex organic molecules. Not 2 of the 4. All 4. What these molecules actually mean: no esters, no cell membrane, no cell, no life as we know it. The esters found in Enceladus's geysers are not a chemical curiosity. They are the structural building blocks of all known biology. Esters make up lipids, fats. And fats make up the membranes of every living cell on Earth. Every cell in your body is surrounded by a lipid membrane that separates its interior from the outside, that controls what enters and what leaves, that maintains the chemical order life requires. That membrane is esters. No esters, no membrane. No membrane, no cell. No cell, no life as we know it. Alkenes are unsaturated hydrocarbons. In industry, we use them to make plastics, fuels, synthetic materials. In prebiotic chemistry, the chemistry that precedes the appearance of life, alkenes are reactive precursors. Molecules from which more complex structures can form if the energy is available and the conditions are right. Ethers. Their presence alongside the esters and alkenes in those same ice grains points to varied active organic chemistry, not an isolated reaction or a contamination artifact. None of these molecules appear spontaneously in cold, inert water. For them to exist together, in this variety freshly ejected from the interior of an ocean, you need conditions: heat, an energy source, organic precursors, and time. Enceladus has all of that. It also has several billion years. This is not life. The Nature Astronomy paper is honest about that, and so should this video be. Complex organic molecules, even direct chemical precursors to the building blocks of life, are not life. The gap between an organic molecule and a living organism, even the simplest one, is enormous. Additional steps are required: the self-assembly of those molecules into replicable structures, the formation of stable membranes, the creation of a mechanism for passing chemical information from one generation to the next. But here is what this discovery changes fundamentally. Until now, the question being asked about Enceladus was, could the conditions for life be present there? Since October 25, the question is, has the active chemistry documented in that ocean had, across the billions of years of its existence, enough time to cross that threshold? Those are two very different questions. The first is hypothetical. The second is a question of history and the history of Enceladus we cannot yet read, because we have not sent the instruments capable of reading it. There is an experiment biologists know well, the Miller-Urey experiment, conducted in 1953. Stanley Miller and Harold Urey filled a flask with the gases believed to make up Earth's early atmosphere: methane, ammonia, hydrogen, water vapor, and ran electrical discharges through the mixture to simulate lightning. After a few weeks, the flask contained amino acids. Amino acids, the basic building blocks of proteins, and proteins are the engines of all biological activity, formed spontaneously from gas and electricity, with no intention, no direction. Just chemistry, under the right conditions for the right length of time. The experiment showed something fundamental. The transition from simple chemistry to complex organic chemistry is not a miracle. It is an almost inevitable consequence of the right conditions held long enough. Enceladus has those conditions. And it has billions of years on any experiment we could replicate in a laboratory. The death of Cassini on September 15, 2017 at 7:55 am Eastern Time, Cassini's signal disappeared from the screens of the control room at NASA's Jet Propulsion Laboratory in Pasadena, California. Not a malfunction. Not an accident. A decision made years in advance, discussed, approved, and deliberately executed. Since 2014, Cassini had been in its end-of-mission phase. Its fuel was running out. Within a few years, without the ability to correct its trajectory, it would drift freely around Saturn until a random collision with one of its moons. The risk was precise and documented. If Cassini crashed onto Enceladus, the Earth microorganisms potentially present on the spacecraft, bacteria that survive in gaskets, in cables, and the gaps of electronic components, resistant to vacuum and radiation, would contaminate an environment that might already have its own native life, making it impossible forever to distinguish Enceladus's own biology from biology we had brought there ourselves. The 1967 Outer Space Treaty and planetary protection protocols applied. You do not contaminate a potentially habitable environment, even if it costs a spacecraft. So NASA directed Cassini towards Saturn itself. 22 final orbits, each one guiding between the innermost ring and Saturn's atmosphere through a corridor 1,500 miles wide that nothing had ever passed through before. A grand finale for a spacecraft that had spent 13 years orbiting Saturn. And on the final pass, instead of pulling back out, Cassini kept going. It dove into the atmosphere at 76,000 miles per hour, and friction tore it apart. In Pasadena, engineers who had spent 20 years with that spacecraft watched the data stop in real time, knowing that Cassini had already been dead for 83 minutes, the time it takes the signal to travel 870 million miles. They were watching the delayed confirmation of a death that had already happened. Some of them were crying. What they did not know that evening? Inside the mission archives, inside the data from 23 Enceladus flybys recorded across those 13 years, were answers to questions nobody had yet asked. Cassini had collected evidence of complex organic chemistry inside those geysers. The instruments had captured everything. The files were there, accessible. It would take 17 years and analysis algorithms that the 2008 team didn't have to read what Cassini had written. The next step is a dedicated mission. The European Space Agency is developing a mission concept to return to Enceladus, not a flyby. a long-duration analysis mission with instruments designed specifically to detect not just organic molecules, but biological structures, proteins, amino acids, sequences that resemble genetic information. If that mission confirms what Cassini's data suggests, it will be the most important discovery the human species has ever made. Cassini was sacrificed to protect a life that had not yet been confirmed. And it was the right decision, because the data it collected before it died suggests there may be something there worth protecting. What makes this story different from every other story in space exploration is this: we did the right thing without knowing it. We followed a precautionary protocol without yet having the evidence that fully justified it. And 17 years later, the evidence arrived. Not from a new mission, not from a different spacecraft, from Cassini itself, from files Cassini created before we killed it to protect what those files would ultimately reveal. That is the real story of Cassini, not the end. The return. Introduction: there is an unwritten rule in space research. The most important discoveries don't come from the moment you make them. They come from the moment someone decides to look a second time. In 2008, a spacecraft flew through the geysers of a moon of Saturn at 20 times the speed of a bullet and collected data for a few seconds. Nobody knew exactly what that data contained. The files were archived. The mission continued. Cassini made 18 more passes over Enceladus. Then it died, deliberately sent into Saturn's atmosphere in September 2017 in front of cameras around the world. In October 2005, a research team reopened those same files with analysis tools that didn't exist in 2008. What they found inside was published in Nature Astronomy, and it changed the question being asked about this moon. Before that publication, the question was whether Enceladus, a moon the size of the United Kingdom, covered in ice, with an ocean hidden under 15 miles of ice and hydrothermal vents at the bottom, had the conditions to harbor life. A hypothetical question. After that publication, the question is whether the chemistry actively running at the bottom of that ocean has had, across billions of years, enough time to cross a threshold. A question of history? In this video, we're going to see what Cassini actually collected when it flew through those geysers at 20 times bullet speed. What Enceladus is ejecting into space right now as you're watching? What is happening at the bottom of its ocean under 15 miles of ice, 870 million miles from here? Why a discovery made in 1977 at the bottom of the Pacific completely changed what we look for on this moon? And why the deliberate death of Cassini in 2017 decided to protect an unconfirmed life carries a meaning today that it didn't have that evening in Pasadena. Stay through the last section. What you are going to understand about why they really killed Cassini is going to change the way you look at what the data revealed afterward.